Research and development in pharmaceutical industry remains a complex, long, and uncertain activity with important costs and rare successes. Dr. White is a researcher working for many years on a genetic disease. Unfortunately, despite some progresses, no efficient treatment is so far available. To understand the complexity of his research, let's remind some biological basis. We are all from a single cell. This stem cell ran through divisions and differentiation processes during development to create all our different cell types. Thus, all our cells share the same genes from the initial stem cell. However, they don't activate the same combination of genes, which defines their genetic state. Their different genetic state defines their specific cell type and behavior. How these diversity of genetic state occurs during development. It is caused by interactions between genes that form a genetic network. During development, cells are submitted to specific stimulations that activate or inhibit genes that in turn activate or repress other genes. All the cells have the same genetic network, but they all have a different genetic state. Now, let's see what happens with a single mutation responsible of the genetic disease studied by Dr. White. This single mutation can create new interactions or repress existing ones. During development, several genes that are not mutated will be impacted by the mutation because of the genetic network. The problem is that Dr. White has only access to the genetic state of cells from experimental data, not to interactions. He doesn't understand how a single mutation can affect other genes. Paradoxically, he's drowning in a sea of data. The challenge is to step from information to knowledge, which unfortunately is not achieved by statistical approaches which are limited to correlations. Vidium tackles this challenge using proven methods from aerospace to master complex systems. The first step is to reconstruct a mathematical model, called in silico model, which is a copy of the genetic network, as a map is for the territory. Our reconstruction algorithm is based on aerospace engineering know-how and biological expertise in stochastic cell behavior. We use the full potential of cloud computing and machine learning to reconstruct unraveled realistic genetic network models from experimental data. Our in silico models of genetic networks reproduce normal development and pathological development due to a mutation. What can we do with these in silico models of genetic networks? We can make diagnostic to identify which portion of the genetic network is perturbed by the mutation. We just have to compare reconstructed genetic networks in normal and mutated development and find new or missing interactions, and thus predict first impacted genes to make reliable early diagnostics. We can identify therapeutic targets and find detour ways to reach a normal genetic state. To change the genetic state from pathological to healthy, we can identify the optimal set of genetic targets to be stimulate or inhibit. We will certainly need to refine the genetic network around the therapeutic targets to propose robust and efficient treatments. To do so, we can predict which will be the most informative new experiment to perform. We then integrate new experiments data to refine the network and find new intermediaries which are new potential targets. Once we know where we should pass, we can predict the best combination of drugs to get there. Finally, we can prepare and follow up clinical trials. We can choose the best responsive patient, monitor clinical result. We can also provide in silico evidences for regulatory approval to reduce clinical trial costs. With all these services, we provide an optimal, secured and rational approach which enables gain of knowledge, time, and money. We propose robust solutions with higher success rate. We support decision-making for development and certification of new treatments.